Hello and welcome to the biology classes. Friends, our discussion was with photosynthesis. So we have come across a long way of photosynthesis wherein we have discussed in the first episode the role of carbon dioxide, the role of sunlight and role of H2O. And then the second episode, little bit of chloroplast we have described. In the chloroplast we have seen that two photosystem occurs, that is photosystem 2 and photosystem 1, which produces two important things. That is, two important components are being produced. To name them, I am writing it is NADPH. It was produced, and ATP. So we have produced in the light phase these two highly energized components because it contains full of energy. How it was produced? That is, in the light dependent reaction, these two things are produced. Well, I should not remind that, because you know that in PS2 and PS1, in PS2 and PS1, that electron transport system was there, that electron pass on here also the electron goes, and here from this ATP and NADP have been produced. Now we will start in the next phase. Next phase is known as light independent reaction. What is that? Light independent reaction. Or phase. Light independent phase. So in the light independent phase, it is very simple and very easy, very comprehensive. Once you want to know a simple thing, I will write this. Simple things. You have to take 6 RuBP. So instead of 6, I am writing here. You should not write this in the exam hall, but for the purpose of your convenience, you write 6 pieces. 6 pieces of ribulose biphosphate we have to take. And again we have to take 6 pieces of CO2. Why I am writing these 6 pieces, later on we will understand. These 6 pieces of ribulose diphosphate having 5 carbon, and it has 1 carbon in each. So I have taken 6 pieces of RuBP. It's known as ribulose bisphosphate. Previously we used to call it as ribulose diphosphate. And coming. And here 1 piece of CO2 contains 1 carbon. So I will write these two comes to produce, this is 5, this is 1 6 carbon compound. So a 6 carbon compound is not available, or it is not stable, a 6 carbon compound. So what it immediately, a 6 carbon compound, 1 2 3 4 5 6, 5 C here, 1 C here. 6 carbon compound is not stable, it is unstable. Now immediately we break up into 2 molecules, 2 3 carbon molecules. These are called PGA, known as phosphoglyceric How many pieces? Here 6 and 6, 12. Here it is 6 and again 12 pieces of PGA will be produced. Twelve pieces of phosphoglyceric acid will be produced. Clear up to this? So I have taken 6 pieces of RuBP, 6 pieces of carbon dioxide, and which unites to form a compound. It is unstable. Immediately 12 pieces of PGA having 3 carbon. Each PGA contains 3 carbon. 12 pieces of this is produced. Now these 12 pieces are not active in nature. You have to activate these 2. Simply we will take NADP and ATP here. From 12 pieces of PGA, phosphoglyceric acid, 12 pieces of PGA, phosphoglyceraldehyde will be produced. Each contains 3 carbon. So carbon for carbon A, 6 pieces of RuBP contains 5 6 are 30 carbon. I am writing here 5 6 are 30 carbon. 30 C. And here 6 carbon. So how many carbons are there? 30 plus 6, 36. Here 12 pieces of PGA, each contain 3 carbon. 3 12 are 36. Here 36 carbon compound. Okay? These are the same because it got NADP and ATP to get themselves activated. Now it has got the power. I have activated this. So these also have 36 carbon. Okay? As simple as that. This 36 pieces, out of 36 pieces, to continue the cycle, I require 6 pieces of RuBP. So 6 pieces of RuBP contains 30 carbon. And here 12 pieces of PGAL, 3 12 are 36 carbon is there, and I have to take 30 carbon. So if I take 30 carbon from here, 6 carbon left, these 30 carbon will constitute how many pieces of RuBP? 30 carbon will produce 6. So I will take 30 carbon, that is out of 12 I will take 10 pieces. 10 3s are 30. So 30 10 pieces of PGA, phosphoglyceraldehyde I will take. So 10 pieces of PGA, each containing 3 carbon, 3 10 are 30. So 30 carbon will be there. These 30 carbon will produce 6 pieces of RuBP. In this way, the cycle goes on. Again, here we have left over with 6 carbon. This 6 carbon will produce, that is 12. Out of 12 I have taken 10 pieces. So I will take another 2 pieces here. Another 2 pieces left. 2 PGAL. 2 PGAL having 3 carbon each. 1 2 3 and again 1 2 3. This will join to produce a 6 carbon compound known as C6H12O6. Okay. So this is the Kelvin cycle. A person, scientist known as Kelvin, actually it is known as Benjamin Kelvin, so it is known as Kelvin cycle. Those who wants to know up to this, for their convenience, because if you are given number 2 or number 3 in the question, then you will get 3 marks, you can write this. Here, this NADPH will come and it will be converted to NADP, and ATP will give one phosphate and will produce ADP. Here H will be there and here phosphate ADP, adenosine triphosphate, one inorganic phosphate will be given. To charge this phosphoglyceric acid to make it active like 12. This is the total funda of this Kelvin cycle or dark reaction, whatever it is. Now what we can observe, what we are observing, that this is light independent phase. No light is necessary. So we can conclude that light independent phase is not dependent on light. But light dependent independent phase is dependent on the products of the light dependent reaction. That is, these two products are required. So indirectly we can say that light independent phase is indirectly dependent on light dependent phase. As or because the two important components which are being produced in phase light phase, that is NADPH and ATP, are required to continue the process of photosynthesis and for the production of glucose or to continue the cycle. So light independent phase is indirectly dependent on light dependent phase. This much you will say, and here again I am repeating, 6 pieces, 6 5 30, 6 pieces, 6 carbon, 36 carbon, it produces a 12 pieces of phosphoglyceric acid, each having 3 12 36 carbon is there. Out of 36 carbon, again it becomes 36 carbon, but it becomes energized with the help of NADPH and ATP. So this NADPH and ATP will produce 12 pieces of PGA, phosphoglyceric acid. Out of 12, 2 will be there, 2 will be left, that is 336, there 36 will produce a glucose molecule, and to continue the cycle, 10 pieces of PGA will come here and it will ultimately produce ribose bisphosphate. Okay, carbon, Kelvin cycle is over, photosynthesis is over. Now, let us make it a little bit complex. Those who wants to, if the number is 2 or 3 in your question paper, 2 or 3 they give you number, you write this. If they give you number 6 or number 8, you have to elaborate it. But main thing you have to keep it in mind. This thing you have to keep it in mind so as to elaborate the next one. So I am erasing this one and let us start with the detail of this. Clear? You take this one, take a pause and write this one in your notebook. We will be studying in the next one. You keep this in mind that this is the shortest form of Kelvin cycle and shortest form of glucose synthesis. Now we will elaborate it. Okay. Let us elaborate. 6 pieces of RuBP. So I am writing 6 pieces here. 1, 2, 3, 4, 5, 6. One, two, three, 4, 5, 6. Each containing how much? 1, 2, 3, 4, 5. So I have taken 5 RuBP. It is known as RuBP. Previously we were calling as RuDP, ribulose diphosphate. But scientists have said here is one phosphate, so I am making it P. It is biphosphate or bisphosphate. Here is another P. Previously also, when we did it, we used to call it RuD-phosphate or ribulose diphosphate. It is all the same, but scientists have said that if this 2 phosphate group consecutively stays in 2 consecutive carbon, then you can call it ribulose diphosphate. But if it is in one position and if it is in 6, 1, 2, 3, 4, 5th position, so it should be called ribulose 1,5, ribulose 1,5. You are mentioning the position of the carbon. Ribulose 1,5 bisphosphate. Bisphosphate. So to differentiate the diphosphate with that triphosphate, we are writing in this way. Adenosine diphosphate, adenosine triphosphate, 2, 3 consecutive. So this is the ribulose biphosphate having 5, 1, 2, 3, 4, 5, 6. 6 pieces I have taken. So 6 pieces of I have taken. Now I will take 6 pieces of carbon dioxide. Carbon dioxide 1, 2, 3, 4, 5, 6. 6 pieces of, I am saying why pieces I am saying to take it very simply so that you will never forget to write like that. So these carbon are like this. So I will join them together to form a product. What will be there? So I have to write it again. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. It is becoming a little bit boring, but it will be interesting in future. 1, 2, 3, 4, 5. And this carbon is joining here CO2. This is O, this is O. CO2, C is coming. This is a carbon. We are not interested in oxygen. So this carbon fits here. So how many pieces are this thing has been produced? An intermediate. This intermediate is called diphosphorebutol. This here 1, 2, 3, 4, 5, 6 carbon compound. This is a 6 carbon compound. 6 carbon compound. Now how many pieces are there? 1, 2, 3, 4, 5, 6. So 6 pieces of 6 carbon compound. It is an intermediate. Unstable. What should I write? Unstable. Intermediate unstable 6 carbon compound has been formed. Since it is unstable, you cannot test it. You cannot test this thing in the laboratory that what is there, because it immediately gets converted to 2, 3 carbon compound. 1, 2, 1, 2, 1, 2, 1, 2, 1, 2, 1, 2. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. So 12 pieces of this 3 carbon compound is known as PGA. Phosphoglyceric acid. That is, it got phosphate because here is phosphate. So 2 phosphate, 1 phosphate will go here, 1 phosphate will be here. 1 phosphate here, 1 phosphate here. So each will get phosphate. So these 3 are called glyceric acid. Now you can say one thing, that out of this 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 pieces, 6 pieces contains the carbon of carbon dioxide. Is it? So 6 pieces, 1, 2, 3, 4, 5, 6 pieces will got this carbon, and 6 pieces will not get. 1, 2, 3, 4, 5, 6. These are all 3 carbon compound. 1, 2, 3. One, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So 12 pieces of PGA has been produced. Out of the 12 pieces, 6 pieces have the carbon of carbon dioxide. Now you can say that the gaseous carbon has been fixed in a solid form, chemical form. So it is known as carbon fixation. So scientists are not accepting this, because scientists were not able to, because Kelvin was not able to explain it. So he has to go through this, and he has said yes, by this carbon is a 14C isotope of carbon, 14C. It makes no difference with this carbon and that carbon, but he has proved that this 14C isotope of carbon, is available in the 12 pieces, in the 6 pieces, out of the 12 pieces, 6 pieces PGA. So it has been conclusively proved that the carbon has been fixed. So this stage is known as carbon fixation. And hence, to give a reward, we have given in 1963, we have given Kelvin the Nobel Prize. So he has become Nobel laureate because he has proved it. In science, you have to prove it. So it has been proved that 12 PGA will be there. And here, phosphate group is there. Somewhere phosphate group is there. So, phosphoglyceric acid containing the carbon of carbon dioxide. Now what do we do? Out of these 12 pieces, these are not that much active. Now we will have to activate it. What we have said, 12 pieces of PGA will produce 12 PGALE. But it takes two steps. How? In the first step, what happens? In the first step, I will take ATP, adenosine triphosphate I will take, and then we will take NADPH. So here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. I will take 12 ATP. Okay? So 12 ATP I will take. Ultimately it will be 12 ADP. Clear? So again I have to write 12 and one phosphate, adenosine diphosphate. Adenosine, it is converting to the diphosphate. So another phosphate will come. Previously, what phosphate it had? It had one phosphate. So it is getting another phosphate. So we will write 12 pieces of, in the first step. This is known as first step. 12 pieces of 1, 3, should I write it? 1, 3, okay. Di-PGA. 12 pieces of 1, 3 di-PGA will produce. That is, the same 12 pieces will be here and another phosphate will be there. So how much phosphate it is having? 2. Clear? So 1, 3, these are two positions where the phosphate group is attached. Say it is in 1, it is here, it is here 1, 3. Here also it is 1, it is here. It makes no difference whether this is 14C or not that. 14C is for the purpose of it. You are taking distilled water or these are the simple water. No difference that much. So, forgetting that it contains another carbon, it is carbon, but it is not in that sense we will take it. Okay. So now 1, 3 di-PGA will be produced. How many? 12 pieces. I am not writing all these things to take it more. Anyway, in the next step, what we will do? In the next step, we will take 12 NADPH. 12 NADPH. So hydrogen will go and 12 NADP will be produced, and here also that 12 pieces will remain constant. 12 pieces of di-PGA, and then this is known as PGA. 12 pieces of these are all the glycerol, HYDE. 12 pieces of phosphoglyceraldehyde 3-phosphate. Will be produced. Not clear? It is known as glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. How? Because there was 2 phosphate here and you are saying 1 phosphate here. So 1 phosphate should go. 1 phosphate should go. So I am taking 1 phosphate from here. Minus H3PO4. This phosphate group is added here. Again, leaving out. So, glyceraldehyde 3-phosphate THOS PHAP. This is known as G3P. G3P. This is known as phosphoglyceraldehyde 3-phosphate or simply PGA. As per your choice, you can name it. So, these 12 pieces of PGA has been produced. Okay, clear. Now, it got 3 phosphate, that is 1 phosphate is there, and 1 phosphate, this is 12 pieces. Out of these 12 pieces I have taken out. So 1 phosphate is still there. So what we will do, out of 12, 10 I will take. 10 PGA or 10 G3P or 10 glyceraldehyde 3-phosphate. As per your choice, you can call your baby. Okay. Now 10 PGA, while I am taking, each PGA contains how much? 3. Here it is 3, here it is 3, here it is 3. Something coming out, something goes out, forget it. Now 10 PGA, each containing these 3. 10 phosphoglyceraldehyde. What I am saying? Phosphoglyceraldehyde. Here 1 phosphate is there. In each 10 pieces, 10 into 3 are 30. So 31 phosphate is there. Whether it is 1 phosphate, that is monophosphate. Whether this monophosphate will produce diphosphate? No, ribose biphosphate. So another phosphate I am to add. What I will do? 10 pieces will constitute 10 pieces. 3 10 are 30. 30 will be there. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5, 6. 5, 6 are 30. That is 6 pieces. So out of these 6 pieces, I will write here. See minutely. 6 ribose monophosphate will be produced. 6 pieces, 6 pieces of ribose monophosphate will be produced because it got 1 phosphate. So I have to add 1 phosphate more. So in 6 pieces, how much I have to add? 6 ATP. This phosphate will go there and 6 ADP will be produced. So 6 ATP I am inserting 1 phosphate and it becomes ribose BisCO. In between, ribose monophosphate has been produced. It was not there because they are giving me number 3. They are giving me number 6. I am writing, I have to write this one, this one, this one, this one. And if you are given number 9, 5, what do you know about this Kelvin cycle? Then if they give 9 number, you have to be more cautious. Now we have left over 12 pieces. Out of 12 pieces, 10 pieces are done. I have got 2 pieces. 2 pieces of PGA, phosphoglyceraldehyde. This I will get in the next episode. Till then, thank you. Please allow me to go in the next episode because it is long time. Thank you very much.